Okay, hello everyone. So we will continue with the last section now for the chapter 3, or the last chapter, which is carbohydrates. So there are 3 chapters that's covered for our midterm exam. We have the introduction. Of course, we have to review and what you have learned from organic chemistry. The second one, we have the proteins, and then third one, we have the carbohydrates. And the last topic in the previous days or discussion that we had talked about the different kinds of carbohydrates. So we started with the monosaccharides, and then we had the disaccharides last time. And if you check on your LMS, I already uploaded a problem set now for which you can work and which you can study before the exam this Friday, April 8th. Okay, so a part of the coverage will be the last topic, which is polysaccharides. So what are polysaccharides? We've learned from the word poly, no? Poly means many, and saccharides meaning many sugars. It's repeating sugar actually from the monosaccharide. So most carbohydrates found in nature are polysaccharides. They are polymers of medium to high molecular weight. So why they are called polymers? And they are polymers because of the repeating units, no? And in this case, the repeating unit is the monosaccharide. We have learned in proteins. Proteins also is a polymer, no? It's a biological polymers. Any polymers with a repeating unit that can be found in nature, we call that one's biological polymers. So for proteins, the repeating unit is the amino acids. Okay, and for polysaccharides, the repeating unit here are the monosaccharides. So polysaccharides are also called glycans, which differ from each other in the identity of the recurring monosaccharide. So just like in disaccharide, now we can identify the particular type of a disaccharide based from the monomers or monosaccharide that is being linked. So there are 2 monosaccharides being linked to produce a disaccharide. In the case of polysaccharide, we also have to identify, or there is also a specific identity of the recurring monosaccharide units. So there's also the length of their chains. We have there is a certain range now where there is the number of the repeating or recurring monosaccharide units, in this case our monosaccharides. So we can also identify our polysaccharides in the types of bond. Remember, in your disaccharides, you have to know, is it alpha 1,4 glycosidic bond, or is it alpha 1,6 glycosidic bond, or beta bakahan, no? And we have to know what is the first monosaccharide and the second monosaccharide in the disaccharide structure. In the polysaccharide, you also have to know what kind of glycosidic bond is being formed here. So the type of bonds, type of glycosidic bond, which linkage is being formed, the linking units, no? And the degree of branching, you also need to know if it's highly branched or not branched, and that will identify our kind of polysaccharides. So there are 2 kinds of polysaccharides types. Now, the first one is the Homo Polysaccharide. When we say Homo, it contains only one kind of monosaccharides or recurring repeating units. There's only one kind, one type of monosaccharide. In the heterosaccharides, there are 2 or more monosaccharides that's being used to be repeated in the structure of the polysaccharides. So both the homopolysaccharides and the heteropolysaccharides, heterogenic meaning it's a mixture. Hetero meaning there are 2 or more kinds of structures present in the polysaccharide. So your homo or heteropolysaccharides can either be branched or unbranched. So branch meaning straight chain lang siya, no? Structure. Pag branch, it's like you have a straight chain structure, but within the straight chain structure, there is another type of glycosidic bond that's being formed that will be branched out from the structure. So you see here, there's one type of 2 types of monomers here, different colors. Now you have pink and blue here. in a straight chain manner. So this is the unbranched type, but along the straight chain linkage, now there is somewhere that branch out from the linear structure here. So you have this branch type of heteropolysaccharides. So I have already mentioned that. So example of the Homo Polysaccharides is that I have mentioned that they contain only a single type of monomer or a single type of monosaccharide. In this case, our monomer here is monosaccharide. Okay, so some homopolysaccharides usually they serve as storage forms of monosaccharides that are used as fuels. Examples of homopolysaccharides, you have starch and glycogen. Later, we'll look at the structure of starch and glycogen. So I think you're familiar already with starch and glycogen already. Most of the time, we are eating the starch from plant sources. So other homopolysaccharides serves as structural elements in plant cells and animal exoskeletons. So we have cellulose. Now this is from plant sources also, and you can have your chitin. Chitin, you can find that sa mga crabs and shrimps. Those are some of the polysaccharides that you can find. So these are examples of the homopolysaccharides. For heteropolysaccharides, I have mentioned that it contains 2 or more different kinds of monosaccharides. Okay, and usually they also provide extracellular support for organisms of all kingdoms. Okay, so the first kind of polysaccharide that we will explore is the starch. So there are 2 kinds of starch. We have the amylose and amylopectin. So what you can see here is the structure of our amylose. Okay, so here is the non-reducing end sugar. Katong continuous na siya, naka-linkage na siya no, here. And the last end is a reducing sugar, which means that this is in cyclic form, remember, and it can do Newton rotation. The process of Newton rotation, remember that it will be at equilibrium, forming a cyclic form. This case is the pyranose, member being cyclic form, and the linear form, katong fissure. So because open man is yan, it's not bonded. This is our anomeric carbon. This part, you can convert this one into suitable bond O, and then straight chain. No? So if it's a reducing sugar, this means that this end, the aldehyde, can be oxidized into the alcohol form. Okay, remember the amount of reactions of the monosaccharide. So here you have the repeating units of, you have down, up, down here, supposed to be OH, but forms a bond with the previous monomer here. And then you have your carbon 5 and you have your OH forming a bond. Supposedly this is your suitable bond OH, your aldehyde sa imuhang aldose na monosaccharide. And pag rotate siya, forming a cyclic form, you notice here, so mawala sa suitable bond OH, but you will have an anomeric carbon. And if the OH, the suitable bond OH in the rotation, if you remember, is converted into an OH. So the OH can either be down or up. So the OH originally is down, it's called alpha. The OH is up, remember it's beta. So here, originally before it forms a glycosidic bond, before it forms OO linkage, carbon-O, carbon linkage, not the glycosidic bond. Before that one, it has been like an OH, and there's been an OH here in the down portion, and they form a glycosidic bond minus the H2O. So OH here, nawala sa H2O, we form a carbon-O, carbon bond. This is the glycosidic bond. So this is a glucose, another glucose, down, up, down, glucose. Okay, so amylose is a kind of starch which is simply made up of the glucose unit, and it is formed by you can see the type of the bond that's being utilized in the amylose is that you have your alpha 1 and carbon 4 of the second monosaccharide, alpha 1,4 glycosidic bond. Okay, that's what you see. And then what you can see here, and it's made up of glucose. So you have your alpha 1,4 glucose glycosidic bond. So this is the type of monosaccharide that you can see. So starch is a Homo Polysaccharide. When you say Homo Polysaccharide, you can see here in the structure you have the same type of monosaccharide. You have down, up, down here. The same, no, there is no alterations because the monomer that's being repeated, so repeat. Unlike the disaccharide that you only see two monosaccharides and then that's it. Now that's the end of your sugar. But here in this case, you can see that it's more than two. So you have 3, 4, and so on. You just have to indicate how many numbers of this glucose is being repeated. Okay, so you can see this is the structure of your amylose, and you have seen here that it is the energy. You have your starch, it's a Homo Polysaccharide. It contains only glucose monosaccharide units. So we have learned that starch actually is the energy storage polysaccharides in plants. You can find it in plants, no? And if exists glucose enters a plant cell, it is actually converted to starch and stored for later use. So since the monomeric unit of our of our amylose, of our starch, is glucose, when it is present in excess amount in plants, no, it becomes the energy storage polysaccharides in plants. That's why when you eat, it becomes our main source, no, of food, the starch for humans, no, because it is the energy storage polysaccharide. So if excess, as I mentioned, if excess glucose enters a plant cell, it's converted to starch and stored for later use. So it can be used by plant for later use for its metabolic process in plants, but at the same time, we as herbivores, we can also use starch as our source of sugar, where it can also be converted once it's processed. Once we digest starch, it can be digested or converted back to glucose, which is the main sugar that's being utilized in the blood. Because the cell cannot get enough glucose from outside to the cell, it hydrolyzes starch to release glucose. So there are two types of polysaccharides, or glucose rather, polyglucose. Starch is also called polyglucose. Okay, so again, I have mentioned that one, you have the amylose and the amylopectin. So the amylose is a straight chain glucose polymer. It contains 15 to 20% of the starch. And you have the amylopectin. No? So the amylose, let's get red. The amylose contains glucose, usually 15 to 20% of the starch. The starch is a mixture containing amylose and amylopectin. So the second one is you have the amylopectin. Aside from amylose, it contains 80 to 85% of the starch. So in amylose, and we'll look at later in the structure, in amylose, now you have the non-bond structure. The glucose are connected by 1,4 glycosidic bond. You have here. This is a 1,4 glycosidic bond. You have the alpha 1,4 glycosidic bond, which is glucose. Okay, later, some alpha. Okay, so in the amylose, now you have contains glucose units. It's around 300 to 500 monomer units. present. So pag natin makita ang glucose na is usually 300 to you have 300 to 500 monomer units percent. The second one, you have our amylopectin. You can see here in the structure, you have your pyranose, no? And it's a cyclic form. You have here down, up. It's not specified here. It forms a bond here. You have here. And second one, you have down, up. You have down, comes from down of the OH. Okay. Down, up, and another, should have been OH, no? There should be a linkage somewhere here. Another unit, you have down, up, down. So this is also made up of glucose, right? And what you can see here in the structure is that you have a straight chain, straight chain. So you have a 1,4 glycosidic bond, carbon 1, carbon 4, no OO. You have a 1,4 glycosidic bond, okay, 1,4 glycosidic bond. And then you have a straight chain, okay, but what happens here is that there is a branching somewhere here. So there is still a branching that occurs between carbon 1 and carbon 6. So supposedly this is our carbon 6, 1, 2, 3. So kung straight chain lang siya, paano na siya? Padayon na siya, no? So that will be if it is an amylose. Straight chain na siya, and you only have, of course, the glucose units here. But when it forms a branching, there is a branching for every alpha-1,6 glycosidic bond. So carbon 1 and carbon 6, this is carbon 6, almost, no? So there is a branching, and then what you can see here is also what you can see here in the structure here, in straight chain, you also have 1,4 glycosidic bond, alpha-1,4, alpha-1,4, alpha-1,4 glycosidic bonds for the straight chain. This is for the branching. So for branching, there is an alpha-1,6 glycosidic bond. So dili siya posibleng nga 1,4 na continuous na siya kung 1,4. So to branch out, dapat na siya alpha-1,6 na glycosidic units. Okay? And so branching also usually occurs for every 25. So pag maabot na ganun siya ika-25 ng glucose, no? So let's say ika-22, 23, 24, 25, or to taman 30, usually 25 to 30, mo branch out na siya, dili siya mo straight chain. So mani ang structure sa amylopectin. Okay, the difference between amylopectin and amylose is that amylopectin, it is a hetero. It's still a Homo Polysaccharide, but there is this branching occurs every 25 to 30 amino glucose units in an alpha-1,6 glycosidic bond. So this is where the branching, but still the main glycosidic bond that's being used is the alpha-1,4 glycosidic bond between glucose and glucose. So because of the branching, amylopectin is a larger average molecular mass than the linear amylose. So mas bugat ang amylopectin, and there is actually up to 100,000 glucose units to be present in an amylopectin polymer chain. So there's a single chain like 100,000 glucose units. But for every, you have to remember that for every, let's say 100,000 na siya, but for every 25 to 30 glucose units, no, na ay branching na nahitabo. You have here. So a straight chain na po, and then with somewhere here it is a branching. So imagine like every 25 to 30 na ay branching na. So dako gid iyahang molecular mass, and this one is a good source again for the energies for a glucose stored, no, in plants also. So amylopectin, okay, have already known that one. So because of that, the amylopectin is a large molecular mass, smaller mass compared to our amylose, kanina straight chain lang siya. The second type of polysaccharides, we have the glycogen. So the glycogen is formed by the, you have here down, up, down, up, down. You have down, up, down, down, up, down. Okay, so this is still alpha-1,4 glycosidic bond, and you have the alpha-1,6 branching in your glycosidic bond. So it's still the same, more or less the same, just like your amylose and amylopectin, no? So like amylopectin, nahitabo lang sa iya ha, na po siya branching, no? Branching niya, so here is a straight chain, but somewhere here there is branching, straight chain, there are branching. Pag branch siya, there is the alpha-1,6 linkage. Okay, but the usual link sa straight chain niya is the alpha-1,4 glycosidic bond. So glucose, alpha-1,4 glycosidic bond. Okay. So the main monomer also for glycogen is down, up, down. You have the glucose also. Okay. So just like your amylopectin, it is branch polysaccharides joined by alpha-1,4 for straight chain glycosidic bond and alpha-1,6 for branching glycosidic bond. So the difference between glycogen and amylopectin is that glycogen is more highly branched. So 8 to 12 glucose units between branches, and the branches are shorter. So remember your amylopectin, branch siya every 25 to 30 straight chain, no? Pag abot niya 25 to 30 or 25, minimum 25 units a straight chain glucose polymer, what happened is mo branch na yun siya. But sa glycogen, ang difference niya pag 8 to 12 glucose units na, mo branch na siya. So mas highly branch ang glycogen kaysa amylopectin. So if you remember, amylopectin, there's already a very high molecular weight. So glycogen, mas grabe ang iyang molecular weight. So another kind of polysaccharide is we have the cellulose. So the cellulose is formed by the alpha-1,4 glycosidic bond still using our glucose. So just be careful when how we look at here, no? Kanisiya is, you notice the normal way of writing katong what we have learned also sa tong monosaccharides and disaccharide is when you have your O here. And this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. You have your O here. And next to carbon O, you have your anomeric carbons. Can either be up or can either be down. But up, before the glycosidic bond, there should be an OH. So it is a beta, right? So kung up siya, it is a beta. Pag down, it is like this one. Pag down, this is an alpha. Okay? But you just have to be careful. Pag i-flip ni mo na, no? Pag i-flip ni mo ni siya, kani sa taas, paabos ni mo, kani sa paabos ni mo. So kung ikan sa up, na siya diri. So si O na diri na si carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So pag diri ang imong O, kani siya is carbon na to, pa-up siya. Ang atong down nga OH, diri instead nga down, up, down. Okay, so up, down, up, baliktad. Down, up, down. So we can also write the same manner as this, no, para di mo magbigo. So just upload it later, no, for a different way. So kung isa ka slides. Okay, you can also write your cellulose in this manner. Ready umagibog. So you can have your just copy. So instead of O, put our O here like this one, and you can just repeat the structure. So alpha-1,4 glycosidic bond, mura siya o sa atong disaccharide, no, but it's more than 2. So it's a repeating unit with alpha-1,4 glycosidic bond. So cellulose is the natural component of plant cells. We have learned that it is the most abundant naturally occurring polysaccharides. And ano siya sa plant cell wall, katong sa mga membranes nato, no? The woody portions of the plant stems, stalks, and trunks have particularly high concentration of the fibrous water cells. Kanang sa plant cell walls lagi, kanang sa gawa sa mga plant cells, no? You can see that one kanang sa plants, no? The woody portion. Stems, kanang sa stems, stalks, and trunks of the kung sa trees, no, or sa any plants made up of cellulose. So like amylose, cellulose is an unbranched glucose polymer. You can see here, it's unbranched. Like, parehas siya kang amylose, katong unbranched. The structural difference between cellulose and amylose, which gives them different properties, is that the glucose residues present in cellulose have a beta configuration. You can find here, this is an beta 1,4 glycosidic bond. In the amylose, you can look at the structure of the amylose. This is an alpha. So parehas siya, 1,4 glycosidic bond, but the difference is we're using here, first monosaccharide is using, or we're using a glucose that is an alpha here for the amylose. For the cellulose, we're using a beta here. So up ang iyahang OH, no, it's a beta structure, and this one also is a beta. So you have here carbon 1, down na yung itsura, no, kaya naman na siya dila, pero baliktaro na yung person. So this kind, the OH is up with the structure. Pag kanina structure, the OH, carbon 1, OH is down. So just think of this, no, kung kanina mong O, 1, 2, okay, this is your carbon 6. Perhaps, yeah, this is your beta glucose, your beta glucose here. So that is the difference between your cellulose and your amylose. Okay, pareha siya unbranched, but in the cellulose, we're using the beta 1,4 linkage rather than the alpha 1,4 linkage present in our amylose structure. Okay, so you can still find that in plant cell walls. Si amylose, na ito makita si amylose. You can still find that one in plants, no? Ang source na nato si amylose, you can find that in plants, amylopectin in plants, cellulose in plants. Glycogen is an energy reserve carbohydrates for animals. So we are part of the animal kingdom, the humans. So we have kanang mga stored source nato sa glucose nato, kanang sa itong mga fats. It's actually, you have the glycogen, no? That's for animals. Like, as we don't have cellulose or amylose starch, no, but we have stored glucose in the form of glycogen. Okay, so cellulose chains contain usually about 5,000 glucose units, which gives maximal cellular masses about 90,000 atomic mass units. It's very, very, because the branching occurs every wala branching din cellulose. Wala branching in cellulose. So usually you have cotton is almost pure cellulose. Example of our cellulose, no, kanang cotton, kining gamit niya sa gamit mo mga stringent. You have your cellulose, kasi yung unlands, also cotton. Also for some of our textiles, no, it contains cotton. Sa cotton is almost purely cellulose, 95%, and wood is about 50% cellulose. Another kind of polysaccharide is you have the chitin. No, here we have the structure of a crab, exoskeleton crabs and other arthropods like shrimps, you mentioned, or other arthropods, kanang insects. No, so in the structure, it's very, very hard. So in structure, no, kung kaano galimang and tuktok na ninyo, it's made up of chitin. So chitin is still made up of this is the structure where your cellulose. So you notice, I should have included this one here. And I'll put it here for the label, with Howard's structure. Still cellulose. So you can see here the alpha 1,4 glycosidic bond, beta, beta, not alpha, beta, so it's up, down, beta 1,4 glycosidic bond. So managkiinanan niya para maklaro ang straight chain. Kapasaka man. So if you use the Howard projection, you can also use this one using the chair. Yung baliktaran pa rin mag-ligo. Okay, so this is your cellulose. So let's go back again to chitin. This is the structure of your chitin. What you can find here is that parehas iyahang linkage sa cellulose. It's a beta-1 for glycosidic bond of our monosaccharide. But our monosaccharide that you can see here is not similar to our cellulose. Like this one, you have your carbon 1, 2, and carbon 2, you have your OH, 3, 4, 5, 6. So it's OH, in the monosaccharide glucose. What you can find here is that ang OH na to is react niya first to NH2, no? There is a reagent, remember your monosaccharide, so we converted that to NH2, and then the acetylates you have here. Okay, so what is our monosaccharide? Our monosaccharide actually in chitin is the N-acetyl amino acid. So I will highlight, it's an N-acetyl, amino acid, N-acetyl amino, which is a derivative of glucose. So N-acetyl amino, derivative of glucose. Okay, so you have that. And that is our repeating unit for chitin. So na siya diri nga part. N-acetyl bond O acetate. Or C-double bond OCH3 as N-acetyl amino derivative. You have your amino here. C-double bond O is an amide bond here and CH3. Okay. So kanina replace na niya siyang OH on the carbon 2 with this structure. And pag ma-replace na siya, then you have your monomer. This is our monomer. This is a derivative of your glucose. If you remember our reaction sa itong monosaccharide, now you have this one. Okay, ito ang sa kong isa ko slides. Napa-isa ka reaction na diri na upload. Okay, anyway. But I have shown that actually before, no, that reaction where you form this, in the other slides, I have shown that reaction here. I'll just upload the other, or insert na